Okay, again, grade 11, neo-Mendelian genetics or post-Mendelian genetics. The third aspect, that is the example, that's multiple allelism. We have seen incomplete dominance, then codominance, then multiple allelism. Let's see. It is generally accepted that genes has two, a gene has two alternative forms. Alleles, as far as Mendelian genetics is concerned, just one gene is represented by two different forms or alleles, two forms of a gene, alleles. This is known as dualism, Mendelian dualism. In his experiment on piece of Pisum Gregor Mendel encountered this phenomenon when one gene is represented by two different alleles. This is Mendelian genetics. In this case, one of the two alleles of a gene is dominant over the other, which is recessive, which is known as complete dominance. But in many cases, several alleles, more than two alleles of a single gene governing the concerned trait is available and is known as multiple allelism. Therefore, how can you define multiple allelism? It is a phenomenon in genetics where one gene is represented by more than two alleles. Typical example of multiple allelism that it comes to our mind immediately is human ABO blood group system. And this is three alleles represent this genotype, three alleles, the IA, IB, and IO. 6 possible genotypes are there: IAIA, IAIO, IBIB, IBIO, IAIB, IOIO, or simply you can write as it is described here, I small, it's possible. Even you can replace this IAIA by AA, IAIO by AO, IBIB by BB, IBIIO by BO, and it's possible. Therefore, 6 possible genotypes, 4 phenotypes. 4 phenotypes are A, B, AB, and O. Human RH and other blood groups. This is also a case of what? Multiple allelism, the RH. This is also based on the antigen, RH antigen on the surface of red blood cells. Coat color in rabbits also showing what? Multiple allelism. Different alleles determine black, brown, or white fur color. Eye color in Drosophila. Drosophila melanogaster is an insect, common fruit fly. Multiple alleles control variations in body color. Multiple allelism in the human blood group or genotype. You can see both blood group systems, the ABO and RH factor genetics, show multiple allelism, I've told you. And Nobel laureate Carl Landsteiner was involved, the American Nobel laureate, was involved in the discovery of both the ABO and RH blood groups. The differences in the human blood are due to the presence or absence of certain protein molecules called antigens and antibodies in the plasma. That means antigens on the cell membrane of red blood cells, antibodies in the plasma. If you have A type of blood, your red blood cell contains antigen A, but your plasma contains anti-B. It will not allow B type of blood to come into your system. If you are given your blood type A, if you are given blood type B, or anti-B antibodies will fight for you there, trying to clump together, mass together, attach each other together, the B blood types which are not part of your blood system, considered as foreign or as unwelcoming agents in your body. The antigens are located on the surface of the red blood cells. Antibodies are in the blood plasma. And the blood group you belong to depends on what you have inherited from your parents, and the gene that determines the type of blood as A, B, AB, or O is called isoagglutinin gene and is represented by 3 different alleles, IA, IB, IO. And Thus, there are 6 possible genotypes to give the 4 phenotypes. These are, of course, repetition or redundancy. These are the genotypes, 6 genotypes, and the phenotypes you can see. AA, the same, 1 phenotype. BB, 1 phenotype, B type. AB, 1 phenotype, 4 different phenotypes, of course. You call this one homozygous A, this one heterozygous A, this one homozygous B, this one heterozygous B. And notably, it says to find the number of different genotype in multiple analysis, the formula is n times n plus 1 over 2, where n is the number of alleles. For example, what are the number of alleles for ABO blood group? 3. Enter 3. 3 times 3 plus 1, 3 times 4 over 2, 6. Yes, we have 6 genotypes. IAIA, IAIO, IBIB, IBIO, IAIB, IOIO, the 6 genotypes. Therefore, to get the number of different genotypes, this is the formula. For example, if you have 4 alleles determining one character, the number of possible genotypes will be 4 times 4 plus 1 over 2. That is 10 genotypes are possible. This is example ABO blood grouping. Therefore, blood group A, if you belong to the blood group A, you have A antigen on the surface of your red blood cells and B antibodies in your plasma. Blood group P, if you belong to the blood group P, you have B antigen on the surface of red blood cell and A antigen in your plasma. If you belong to blood group AB, you have both antigens, antigen B and antigen A. If you belong to blood group O, but blood group AB has no antibodies in the plasma, that's why it can receive any type of blood from any donor. O type is opposite to A. It has no antigens but has both antibodies, anti-A and anti-B. Therefore, it can't accept blood except from its own type. This is a table showing the different genotypes, the antigens, antibody made by body. Therefore, this is redundancy, of course, repetition. The second type of multiple allelism is Rh blood group, of course. Many people also have so-called Rh factor on the red blood cell surface. This is also an antigen, and those who have it are called Rh plus. Those which do not have this Rh factor or antigen are known as Rh minus. Therefore, those which are who are Rh plus do not have anti-Rh antibodies. Those with Rh minus do have anti-Rh antibodies in their plasma. Therefore, if you give Rh minus, if you give Rh plus to Rh minus individual, there will be agglutination. It will not accept it. Therefore, you can give Rh minus to Rh minus. You can give Rh minus to Rh plus, but you can't give Rh plus to Rh minus. But a person with Rh minus blood can develop Rh antibodies in the blood plasma if he or she receives blood from a person with Rh plus blood. Those Rh antigens can trigger the production of Rh antibodies and agglutination is there. Therefore, you don't give Rh plus blood to Rh minus individuals. When your blood type is told you as A plus, B minus, O plus, the plus and minus are because of this Rh factor. Therefore, if you are told you are B plus, your blood type is B, and you have the Rh factor also. Rh factor. If somebody's blood type is A minus, the blood type is A, and he lacks the Rh factor in the red blood cell. A person with Rh plus blood can receive blood from a person with Rh minus blood without any problem. Rh factor and pregnancy. Blood is frequently exchanged between mother and the fetus during childbirth. Thus, Rh negative mothers often immunized by blood from Rh positive fetus if Rh minus mothers have Rh positive babies inside their body, fetuses, which may result when there is Rh positive, when the father is Rh positive. You see, when Rh negative woman marries Rh positive man, they can have a baby with Rh positive blood type. Since there is exchange of frequent blood components can be exchanged through placenta, of course. The Rh negative mothers immune system recognizes the Rh positive coming from the womb, the uterus, from the fetus in the uterus. Usually, no ill effects are associated with exposure of the mother to the Rh minus positive, Rh positive antigen during the first childbirth. The first baby may be born without problem, but after the first baby, the next babies, subsequent pregnancies, Rh positive children or babies carried by the same mother, however, exposed to the antibodies produced by the mother against Rh antigen. Therefore, the mother's body now, for the second, the third, the fourth pregnancies, the mother's body is now ready building anti-Rh antibodies. And these antibodies cross the placenta and start to attack the fetus or the baby and finally kill it. They can kill it. And the mother may give birth to dead baby. This is known as the Rh disease or hemolytic disease of the newborn, they call it. Hemolytic disease of the newborn. Or it can be also called hemolytic jaundice, of course, or hemolytic anemia. Erythroblastosis fetalis is also the name. It can result in death, ultimately. Rh incompatibility. The other NeoMendelian genetics is epistasis. Epistasis is the interaction between genes, where one gene masks or modifies the phenotypic expression of another gene. These genes, different genes, you see, these genes could be located on the same chromosome or different chromosomes. Epistasis is also studied after Gregor Mendel. It's part of NeoMendelian genetics, of course. Epistasis occurs when the effect of one gene, the epistatic gene, overrides or alters the effect of another gene, the hypostatic gene, at a different locus. Meaning the expression of one gene depends on the presence or absence of another gene in this case. The other aspect of NeoMendelian genetics is polygenic inheritance, of course. Polygenic inheritance describes traits that are influenced by two or more gene pairs, leading to a continuous range of phenotypes. This is a result of continuous variation, of course. Polygenic inheritance causes continuous variation, a continuous range of phenotypes. You see, many genes are working on one character, and these many genes are located on the same chromosome or different chromosomes. And polygenic inheritance is not seen in Gregor Mendel's experiment. All Mendelian genes are monogenic, one gene controlling one character. And therefore, this is what? This is the aspect of NeoMendelian genetics. For example, traits such as height, skin color, intelligence in humans are determined by multiple genes, each contributing to the overall phenotype. Okay? And this is all about non-Mendelian, NeoMendelian genetics. Thank you.